In Chapter 7, we move on to talking about the specific genes that are located on the X and Y chromosomes, that is to say, those genes that are located on our sex chromosomes. So we're going to talk about inheritance patterns of the sex chromosomes. We're going to talk about what happens when you have errors that lead to incorrect numbers of sex chromosomes. And we're also going to talk about the ways that sex is determined in different groups of organisms. And then we're going to talk about dosage compensation, which has to do with the number of sex chromosomes that individuals receive. So in general, species exhibit sexual dimorphism. So this is physical differences between males and females. And related to that, sex is determined by the presence of specific sex chromosomes. In the case of humans and many other mammals, we have the XY complement of chromosomes. We'll talk about some different ones a little bit later. And we will talk about the behavior of genes that are located on the X chromosome. There are also genes located on the Y chromosome, so we'll talk about those as well. All right, so sexual dimorphism is essentially just differences between two sexes. These are physical characteristics, sometimes biochemical characteristics, differences in anatomy, physiology, coloration, behavior that separates or is characterized in the different sexes. So there's some examples here on the right. You have elk with these big horns. Many ungulates have those, those are not horns, those are antlers, have those big antlers. Their coloration's a little bit different than the females in the back. Males are often larger than females, not in all cases, but often the males tend to be a little bit larger. In birds, you get flashy coloring in males to attract the females. And then down here on the bottom, we have lions. So you know that the male lion looks a bit different than the females. And most of these physical characteristics are actually ways that the male attracts the female. So these are sort of mating differences, characteristics that help to attract the opposite sex. This is called sexual selection. But all of that is based in the presence of sex chromosomes. That's not to say that all of the differences in males and females are encoded on those sex chromosomes. In fact, most of them are encoded other places. But it is the presence of those X and Y chromosomes that help determine the sex of an individual. So we're going to sort of focus on the XXXY complement of chromosomes. There are other mechanisms for determining sex, which we'll talk about in a later video for this chapter. All right, so here we have a human karyotype. This is here just to show what the sex chromosomes look like. So we have 23 sets of chromosomes, two homologous copies, one from each parent. So on the top we have a female. You'll see that this female has two copies of each type of chromosome, numbers 1 through 22. Those are the autosomes. So these are the non-sex chromosomes. And then you'll see there in the right-hand corner of that top box, we have two X chromosomes. In humans and many other mammals, females are females have two X chromosomes in all of their cells, and it's the presence of those two X chromosomes that make them female. On the bottom here, we have a male karyotype. Now this one just looks different because it's a different type of staining, but the chromosomes are much similar, so very similar. So if you look at the size and shapes of all of them, you'll see that they're the same, except for those X and Y chromosomes down at the bottom. Now males have one X and one Y chromosome. So we're going to talk about the differences, why females have two X's and why males have an XY, and what that means for the sex of an individual. So let's start out with some terminology here. Don't let this scare you too much. It's not too bad. In females that are XX or any other sex chromosome complement where you have two of the same sex chromosomes are called the homogametic sex. So again, homo means same, gamete means gamete. So each gamete has two of the same type of sex cells. We say that any genes that are found on an X chromosome are X-linked genes. It just means that their location is on the X chromosome. And then males have 2 different sex chromosomes, so we call them the heterogametic sex, meaning just simply that they have 2 different chromosomes. We'll see later that sometimes this is flipped. Sometimes in other species, males are the homogametic and females are the heterogametic. It just depends on the species. Any genes that are found on the Y chromosome are called Y-linked genes. There's just a little typo right there. And then one more term, the X and Y system, because the males have different types of different set of chromosomes than males. Males are also said to be hemizygous for genes on the X chromosome. That just means that because they're XY, they only have 1 copy of all of those X-linked genes. So we say that males are hemizygous. This means that anything that shows up on the X chromosome only has 1 copy. Now, X and Y are only semi-homologous. Remember when we introduced the idea of homologous chromosomes, we said that homologous chromosomes are same shape, same physical characteristics, same size, same gene locations. Well, if you look at those X and Y chromosomes, you can see that that meets basically none of those characteristics. So they are homologous in the sense that they do pair up in meiosis, but they have very few genes in common. They do have a few genes that sort of help the homologs line up. But they really don't have too much in common. There are very few genes basically no genes on the X chromosome that are also present on the Y. And any regions that you have similar are going to be just sequence similar, not necessarily coding genes. So the first sex chromosome studies that sort of actually allowed us to see what genes were located on them were done by Thomas Hunt Morgan, and he used the fruit fly Drosophila, and he was primarily working, when he was working on sex chromosomes, he was primarily working on something called the white eye mutant. Now, we saw this white eye mutant in our lab when we did our fruit fly lab. You might recall that fruit flies have either that pretty bright red eye color or a white eye color, no pigment in their eye. Now, this work was really important because it's sort of a foundational experiment in genetics. Thomas Hunt Morgan was already working with fruit flies. He was looking at fruit fly mutants, and his work on fruit flies and the role that sex chromosomes play in heredity actually earned him the Nobel Prize in 1933 for physiology and medicine. So Morgan's work is really important because it told us a few things. It told us that some traits are sex-linked. That means that some traits are found on the X chromosome. And therefore, those traits are carried on either the X or the Y chromosome. So this led him to conclude that other genes were probably carried on specific chromosomes as well. It made sense that if you could pinpoint a gene on one chromosome, that there were going to be other genes on other chromosomes. Now, more specifically, he gave the white eye color a location on a specific chromosome. So he was able to give a very specific gene a very specific location on one of the sex chromosomes. So let's look at what Thomas Hunt Morgan was doing. So he was going through these mutants, and one day he came across a single male fly that had white eyes. Now, this would have come up from a spontaneous mutation. So we talked about how mutations come up in a previous chapter. So this would have been a spontaneous mutation that was not previously present in the population. So some notations here, we are introducing using X and Y, or actual chromosome labels here. Now, when Morgan was working on this, he didn't realize at first that these genes were located on the X and Y chromosome. So we're sort of applying the X and Y chromosome here because we know now that it's applicable. He didn't know what was applicable. He just knew that he was looking at a new mutation. He didn't know where it was. But we're going to use the X and Y so that we can keep track of what's going on here. This W plus is what we call the wild type notation. So this is sort of the common mutation found in the population or found in the wild. Now we use that's not a T, that's a plus, a superscript plus, to indicate the normal version of the gene. And then the mutant version is just going to be W for white eye. So this is telling us that we are looking at the white eye mutant and the normal version of what would be the white eye. So in this cross, he crossed sort of a normal, quote unquote, normal female that he knew was wild type. Basically, he knew they were wild type because he had been breeding them this way. He knew that they were always going to have red eyes. And if this white eye was a spontaneous mutant, then none of these other flies would have it yet. So we start with the red-eyed female, and he crossed it with this white-eyed male. Now, when he did this, he discovered exactly what we would expect to see from a Mendelian cross, where the next generation has red eyes, which suggests that this trait is recessive. If you remember that when we hide the trait in the first generation of offspring, it's being covered up, so it is recessive. So let's look at the cross that he actually did. I'm going to write out the Punnett square. So he crossed a white-eyed female with a red-eyed female with a white-eyed male. So just like all of our other Punnett squares, we're going to write this out, remembering, of course, that these represent the potential gametes that could be formed by this pairing. And as expected, we are going to get all red-eyed individuals. Now, all of these individuals have at least one copy of the red eye allele, and so all are red-eyed. So now this is our parental cross. These individuals inside are the F1 generation. So just like Mendel, we're going to cross our F1 generation and see what happens. So here's our F2 generation. Now you'll notice that white eye came back just like we would expect in regular Mendelian inheritance in a 3-to-1 ratio. So we have 3 red eyes to 1 white eye. Now I want you to notice something though. The only individuals that have white eyes are males. Now this is curious. This is not something that they had seen before. This is not a typical Mendelian inheritance pattern because the trait is limited to one sex. It's limited to only males. So let's draw out this cross. So this is our F1 cross. Now remember, all of the females from that cross ended up heterozygous. Now we're going to cross those individuals to a red-eyed male and see what happens. So you'll notice here we have a red-eyed female, a red-eyed female, a red-eyed male, but here in this gamete we get that white-eyed male. So when we write out our ratios for a sex-linked trait, you have to indicate the differences between males and females in your ratios. So we're not just going to say that this is 3 red eyes to 1 white eye. We are going to say that this is 2 red-eyed females to 1 red-eyed, it's not what I wanted to do. So 2 red-eyed females to 1 red-eyed male to 1 white white-eyed male. So very often students don't distinguish between the fact that these are found in different ratios in the sexes. So let's go back here. So one thing that you'll notice on here is that the pattern of inheritance on the males is going to be different than the pattern of inheritance on the females. Now, this is because Thomas Hunt Morgan reasoned that eye color gene must be found on one of the sex chromosomes. Now, because females can, we'll see in just a minute, females actually can have this trait, he hypothesized that it was actually an X-linked gene. Now, X-linked recessive genes specifically are going to behave a little bit differently than autosomal recessive genes. And we'll talk about that in just a second. So now let's look at what happens if we cross a heterozygous female. So when we do this, meaning that she has that white-eyed allele, she's a carrier for that white-eyed allele, you can actually see that we are going to get a white-eyed female, and we actually see that here. Our other female is going to be heterozygous, and then our male, we're going to have one male and one female. I'm sorry, one red-eyed male and one white-eyed male. Now that's because the female is passing on that allele to both of her children, or both sexes in her children. So here we actually have a 1-to-1 to 1-to-1, one red female to one white female to one red male to one white male. And our genotypic ratios are going to be the same as well, 1-to-1 to 1-to-1. So let's summarize what we've seen here. This is what Morgan saw. He found that eye color is related to, meaning inherited with, the sex of the fly. Flies have XX or XY chromosomes, and that genes located on the X chromosome behave differently. Now, the XX in females behaves as usual, meaning that if you have dominant and recessive alleles, they're going to behave just like they did in our Mendelian genetics chapter. If a female has 2 copies of the recessive, you're going to see it, but otherwise you're going to see the dominant trait because that recessive allele is hidden by the other dominant trait. But in XY males, it behaves differently, and that's because males only get 1 copy. That means that they express the recessive trait with only 1 copy. When it is on that X chromosome, they only have 1 copy, and therefore the only thing you can see is that recessive trait. And again, that's because there's no complement or homologous gene on the Y chromosome. Now, this also means that sex-linked traits are more common in males because they only need to get 1 copy of the allele. So when we look at sex-linked inheritance, we actually see a crisscross pattern. We see that females pass their traits on to sons. Now, they can pass it on to daughters, but we see most common passed on to males, and males are going to pass their traits on the female. So in this case, the female is passing on her white-eyed allele. And you can see that in her offspring here, he only has 1 copy and is showing the allele. Males are going to pass on, in this case, the red allele to that female. Now, if this male had a white eye allele, he could, of course, pass it on to his daughter as well. So something else that's important about this is that it's sort of obvious once you say it, but sometimes students don't think about it. Males cannot pass X-linked genes to their sons because males are only passing on that Y chromosome. So females can pass on an X-linked trait to males or female offspring. Males can only pass X-linked traits on to their female offspring. All right, so Morgan's experiment was quite important. He noted, or he was able to link Mendelian inheritance to chromosomal behavior. And from this, we can deduce that those X-linked traits follow the behavior and movement of chromosomes in meiosis. Remember that in meiosis, homologous chromosomes are separated, and that's how we get this pattern of females passing to males or females and males passing to their daughters only. Now, these fly studies were also important because they supported the chromosomal theory of inheritance. Now, this was an idea that was put forth in 1902 by two scientists named Sutton and Bovary. And the chromosomal theory of inheritance said that genes have specific locations on specific chromosomes. And Morgan's work in showing sex-linked eye color was able to support that genes have specific locations on specific chromosomes. So there are quite a few well-known sex-linked disorders. The most common is X-linked colorblindness, the most common type of which is red-green colorblindness. You can also have blue-yellow. And then there's another kind that is called monochromacy, which is complete colorblindness. This one's really rare. But you can see here, X-linked colorblindness occurs in about 8% of males, but only 0.5% of females. Again, that's because males only need one recessive copy in order to show that trait. A couple other well-known examples are Duchenne muscular dystrophy. This is a progressive weakening of muscles. Again, tends to be more common in males. Hemophilia is another one. Hemophilia is an absence of a blood clotting protein, and so you can imagine there are a lot of complications from this, healing from injuries and things like that, that would make this disease quite bad. The hemophilia was made popular because it was found that Queen Victoria of England was a carrier of this, and due to intermarriage of royal families in Europe in the late 1800s, early 1900s, there's a high frequency of hemophilia in European royal families. So let's go through a couple of examples of Punnett squares with this. So let's look at color blindness. So let's call our allele N. So N is going to be normal vision, and lowercase n is going to be colorblind vision. All right, so this is an X-linked trait. So let's say that we have a normal female. This is a relatively low frequency allele, so we're going to assume that we're starting out with a female that does not carry the allele, but she marries a male that is colorblind. So what are we going to see in their children? Well, let's write out our Punnett square. You guys should be pros at this point. So you might predict, because this female is homozygous dominant, that all of the individuals are going to be normal. So we have all normal vision here. All of our females are going to be heterozygotes. So we have one female heterozygote to one hemizygous male with the dominant allele. Now let's say that their daughter, who is heterozygous, so she's heterozygous, let's say that she marries a man and has children with normal vision. So here, let's see what happens. Now, keep in mind that she is a carrier because her father was colorblind. So if we look at daughters, they're all going to be normal again. But when we look at the males, we're going to see that half of the male offspring are going to be colorblind. So again, we have the 1-to-1 to 1-to-1 genotype. For phenotype, we have 2 normal vision females to 1 normal vision male to 1 colorblind male. So there are 2 other combos that I'm just going to leave here for you to practice with. This is a heterozygous female with a colorblind male, or a colorblind female with a normal vision male. So go ahead and try those and do those, just see what you get for practice.